I'm medical oncologist based here in Barcelona and one of the co-chairs of the meeting. And it's an honor to be here and welcome you to this press conference. So we have just finished the plenary session for really important presentations. We have discussed tumor-agnostic tumors, then tumor biomarkers for immunotherapy. We have discussed the possibility of cure in metastatic lung cancer patients, and finally, also importantly, what could be the contribution of artificial intelligence. So now today we are going to highlight one of the most important presentations of today's meeting, and we would like to start with Dr. Govindan. He will present at 10 a phase 1 study of safety, tolerability of a new compound, AMG 510, novel KRASG12C inhibitor.
Thank you. Good morning. We'll be formally presenting the results of this phase 1 study in about 15 minutes, so I'll just go over this. These are my disclosures. And just to set the stage, lung cancer is increasingly seen as a disease with multiple subtypes, including one of the important types, KRAS mutant lung cancer. And there are different kinds of alterations in the KRAS gene, and one of the most common alterations we see in lung cancer is the alteration called G12C, where the KRAS protein gets overactivated and ends up resulting in excessive proliferation of the cancer cells. And that is a fundamental cause of cancer in about 12 to 13% of lung adenocarcinoma. And unfortunately, this particular group has been very difficult to target so far. And a few years ago, there were breakthroughs made in the medicinal chemistry and the biology, and AMG 510 represents one of the first novel, first-in-class small molecule that specifically binds to the mutant protein of the KRAS, as you can see here. And this is the mutant protein. This is the drug AMG 510. It binds to this, the cysteine component of that. It irreversibly locks it in the inactive state so that KRAS activity is restrained. And this is very, very specific and customized for the mutant protein, and therefore we expect fewer normal tissue toxicities. And the highlight of this is a phase 1 study, and I just want to say a few things in the interest of time, that this is a phase 1 study. The very first patient went on this study just about a year ago. And all these results that we accumulated have come on just within a year of research. And we'll be focusing our presentation on the lung cancer cohort. This is a broad-based study, included not only lung cancer, but cancers, colorectal cancers and other cancers. And what we see here is that in a small number of patients, but a fairly good number for a phase 1 study that's gone on only for a year, in 34 patients that are evaluable, and 34 patients that the subject of this talk, and 23 patients were evaluable. 96% had disease control, and more importantly, about half of them had partial responses, which is a hallmark of activity of a drug. And we did 4 different dose levels, and starting with 180 milligram going up to 360, and 720 and 960. And it turned out we decided that 960 milligram is the phase 2 dose. And at that 960 milligram, the evaluable patients, about 13 of them, were looked at as of July 2019, and we can see the response rate of about 54%, and strikingly, 100% had disease control. In other words, nobody had disease progression on the first scan. As you can see here, many of them had a decrease in tumor size. And how durable are the responses? The important thing is about 8 of 11 patients who had partial responses at various dose levels were still on the study. And the same with stable disease. 8 of 11 patients who had stable disease on the first scan continued to be on the study. And as you can see here on this, the partial responses happen fairly quickly, as we come to expect with targeted therapies, and many of them are ongoing. In fact, about 5 patients or so are nearly 6 months or longer on this. One of our own patients with a partial response is doing extremely well going on for more than 8 months. And this is clearly a very encouraging set of data. And other important point here to look at, we saw responses at all different dose levels, but we have the most experience with 960 milligram dose level. And as was expected of the drug design, the toxicities were very minimal. And with all these studies, especially in a sick population like lung cancer, you would expect to see a lot of adverse events. And about 76% had adverse events. Most of them are disease-related. But the most important column is this here. A third of them had adverse events, but mostly they were grade 1 or 2. And nobody had grade 4 toxicities, which is a severe form of toxicities. And these numbers are extremely reassuring. Nobody had dose-limiting toxicities. No patient had serious adverse event, fatal adverse event, or adverse event leading to discontinuation of therapy, showing that this drug works the way it was intended to, very specific for the G12C KRAS mutant disease. In summary, I would say that for a study that has gone on for just about a year with about 34 patients with the early look, these data are very encouraging. The activity is quite striking with 54% response rates, and the early indications are that these responses are probably durable. Only time would tell. The toxicity is extremely minimal. The most common toxicity attributable to the drug seen in a couple of patients were nausea and diarrhea, very easily manageable. And the phase 1 and study is ongoing with expansion cohort, and phase 2 monotherapy is ongoing as well.
Thank you. Any question for Dr. Govindan? No. Thank you.
Thank you.